Log on, tune in, find out. Another good idea from Cambridge.
So thank you very much, Vlad, and for inviting me to be here. Thanks for the organizers. A real pleasure to be in this conference. And I have found myself in my career preemptively apologizing for having too many formulas and Greek letters in presentations, especially to clinicians. But I think it's a first to have to do the reverse. So I have to apologize for not having any formulas. And there are 2 slides in which you're going to find sigma, and that's about it in my presentation. So just concepts, so bear with me. It's a talk that relies a lot on work that has been done by other people, so I'm just going to be in the talking head. And Sujan has mentioned this formal working group. I think there are 5 people from that in different instances of the formal working group that are present in this conference, so it's kind of interesting. And others that are involved indirectly, like Anastasia, so methods that are based on her work. But anyway, so let me move forward because I'm just between you and lunch. So, all right. So the motivation, and it's okay, why are we talking about this, right? So there's like this perception that we need to improve drug development. So I just want to give you like a couple of slides on what's the situation, what's the problem that we're facing, and more specifically, why they need to improve those findings. So we have been talking a lot about that, but so just trying to recognize the problem even further. And then I'll focus as an illustration of one way to do this kind of explorations on this pharma adaptive dose ranging studies working group. And I'll just end with some further thoughts and conclusions. So the motivation for this is what's called a pipeline problem, right? So essentially what companies are facing is that it's getting harder and harder to get approval for a number of reasons, for a perception of greater regulatory scrutiny on safety especially and other considerations, but also because the pipeline itself is kind of drying out. So there are many diseases that have been taken care of, but like it's hard to get like new blockbusters or drugs that are like really clearly excellent. So tough, and as a result, you see a lot of mergers from different companies because companies try to buy pipelines, right? So that's what we get. So we see that things are drying out. We know that well in advance. We say, what can we do? I have a lot of cash, so let me buy another company that has other molecules, but that has a finite number of steps that you can go before you go to one. So there's a very urgent need for better ways of doing drug development. So it's perceived as being very inefficient, and basically the old way of doing it, which is based essentially like empirical, like you just keep trying, and no success, and that finances all the rest, is not sustainable. So a more scientific approach to the development itself, as outlined in the Critical Path document from 2004, is still very much needed. So we do a lot of science in coming up with molecules and trying to come up with how should we treat a certain disease, but when it comes to the development phase, we are not at that same level of science application. So forums like this are very critical. So people working, especially the young people in the audience, trying to have a career in research, that is a very, very fruitful area. So in particular, we have to think about better methods of use the information that we already collect. So there's a lot of information that's collected in clinical trials, like lab data, safety data, efficacy data, longitudinal data, biomarker data, you name it. So those of us involved in clinical development, it's overwhelming, right, the data sets that you collect. But we don't make as good use of it as perhaps we should. So how to connect the different pieces of information, how to relate that. Beyond that, how do we connect across different phases, or across different trials? Though the mindset tends to be pretty much one trial at a time, because in general, you get one team to work on the phase 2B, another one with some overlap, but not that much, on the phase 3, another one on the drug-drug interaction. So the mentality tends to be a bit more of isolated events rather than trying to connect them all. So how to make better use of that coming all together. And finally, which is one of the topics of a lot of the talks that you have seen here, how to have designs that make better use of the information that you collect per patient, which is very valuable, expensive in one way, but also ethically kind of valuable, that we have to be mindful of that. All right, so zeroing a little bit on the dose finding piece, which is the one that I'm going to be talking about. So why, in particular, do we have to improve dose finding? Well, there is a perception, and that's an old slide, so it is from another presentation given by several of us in the past, but it remains pretty much the same. I think I don't have anything to change, that's what I'm using it. So there's this number that's quoted that the FDA reported that drugs approved between 1980 and 99, in this 20-year span, 20% had to be changed afterwards because of wrong dose. So these are drugs that were approved, right? So you're not even talking about doses that did not make it because they had too high a dose, let's say, and some safety problem. And there's a great perception in the industry that has happened many, many times. The drugs that could have been good, that will never see the light of day just because the dose was too high, and no one is going to go back and invest another $50, $100 million to prove that, with a lower dose, I could have gotten perhaps same efficacy and much better safety. Hence, strong interest from FDA and EMA on quantifying this minimum efficacious dose. So, okay, you have a dose that seems to work, the safety in the small patient population that you observe seems to be okay, but can you show me that a smaller dose would not have the same efficacy and supposedly should have better safety? Bottom line is that there's a perception that one of the root causes of the big problem is that we don't get the dose right. I'm speaking as an industry person here. All right. One of the, and again, as a statistician now, one of the perceptions is that a reason for that is that people tend to approach phase 2 trials, which are in essence exploratory and let's say more model-based, as if it were a mini phase 3 trial. So Vlad mentioned about that, and you're going to be seeing that frequently. So they tend to see that, well, how should I be selecting a dose? And they approach that as a hypothesis testing problem. So, well, let me test until I don't have significance, and that's the dose that I'm going to bring forward. All right, probably that's not the most efficient way of doing things. So how to improve? How can we, and again, I'm not talking about the math of how to do this, but more the concept of how it could be done. One is try to move away from this phase 3 view, right? So it's, and again, a lot of the blame for that goes to statisticians. I think we have brainwashed clinicians to say, well, that's how we calculate sample size. We get a hypothesis test, we specify a power, a significance level, we go into n-query or some other piece of software, and that's the sample size that you need, even if that's not the question that you're trying to answer. So we say, forget it. So I know how to answer that question, and that's the question. And clinicians pretend that that's okay. They really say, well, okay, you get me a sample size in the end. I don't need you anyway because I'm going to get in a room, look at all the evidence in the data, get a gestalt, and that's the dose that I'm going to be selecting into phase 3, which, again, it's not a very scientific way of approaching it. So it's, I guess, an agreement of two different professions pretending to be doing the right thing, and at the end, there is a problem that occurs from it. Now, better exploration of the dose range is critical. So it's kind of frustrating to think about methods to do dose finding and model-based, blah, 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 and so we get excited about it, and at the end, well, here's what we have, placebo and 2 doses. And say, well, what kind of a model can I fit to 3 points, right? And that, if you're lucky, sometimes, well, let's just go with 1 dose because we really believe that that's the dose that's going to be the one that's going to work. So you do like this binary search of dose finding, which is not very efficient either. It's not an easy task, however. So examples like Vlad was presenting, that's the exception rather than the rule. So there are many, many reasons why people are going to give to you why they don't want to go with 6 doses and placebo, right? Even if you go adaptive. Another key problem is that there's always the hope that the phase 2 trial is going to be so successful that they can go to FDA or EMA and say, well, this is one of my pivotal trials and I just need another one because look at the effects that I got. And that kind of affects the whole idea of the model. So there's a lot of concern about, like, say, preservation of type 1 error rate, for example. There's well, let's really go the hypothesis testing framework because then we can convert that into pivotal and then we just need another one. But always this hope in the background, which of course it's silly because you end up not doing either the exploration or the pseudo pivotal in a good way. So you sacrifice both, but it's prevailing. Until today, I see a lot of that. Another one is like allocating proper resources. I know Chris's talk was interesting and kind of for me, a little bit counterintuitive in the sense that, well, I always think that we don't invest enough in phase 2, but I understand that it's kind of putting that in the context of the phase 3 may be an interesting way of balancing it. But in general, people want to do that on the cheap, right? So let me do the phase 2 quickly, let me go straight into phase 3, and then I'm going to be investing my resources. And may not, from a decision theoretical approach, maybe not the best way that can be done. So in terms of improving those findings, one thing to take into account, and you've seen that quite a bit in the talks so far, is that it's not a one-factor thing. So we just don't talk about adaptive designs, we just don't talk about optimal designs. It's a combination. A lot of the methods that you have been presented here, they combine either being partly adaptive, maybe based on an optimal design approach. There's going to be a Bayesian component, model-based, and so forth. So when combining all those features together, sometimes it's hard to see that, well, where is the value really coming from, and does it matter? So is the value coming mostly from doing model-based Bayesian, or from adapting, or vice versa? So, and taking that into account and peeling off a little bit may help you allocating resources in a more efficient way. But it doesn't matter what method you use, the evaluation of these operating characteristics is critical, and you have seen over and over examples of people presenting simulations, different scenarios. That's how it works, that's how it doesn't work, and I'll do the same if I have time to do it. Now, there are a number of challenges, and I have mentioned some of them. So, but there's like a broader challenge, which is the mindset. So by and large, the pharma industry tends to be quite conservative, almost by training, right? Because it's a very regulated industry. So the people who succeed in that industry tend to be conservative by nature. So they tend to try to reproduce what they have done in the past because it worked in the past. And that's why they were promoted in the first place, because something that they did worked and that they got the promotion. But you try to change that, it's not easy, right? So people say, well, yeah, that sounds very good, but at the end, I want to go with my 2 doses placebo, done a test, and so forth. Now, one issue that is critical too is that of operational infrastructure. So we can talk a lot about doing lots of adaptations, having lots of doses, which is very appealing when we run the simulations from a theoretical point of view. But sometimes you face a lot of resistance from people in the operational side, which have a very different reward system than yours. So they want to recruit as quickly as possible and be done as quickly as possible. So trying to convince them that slow down recruitment because we need to learn as we go is not an easy task. And those of us who have actually done this kind of work know very well how difficult that might be. There's like more resource needs that are needed as well. So you don't just go there and change a culture overnight and say that, well, let's do Bayesian normal dynamic linear models, and there's not enough people who will be able to do that. There's not enough software, and there's a lot of things that need to be in place for you to do this. And finally, and probably one of the most important, that all of us, because of this pipeline problem, because of the rush to get like things out the door as quickly as we can, are faced with this speed focus. It's all about speed, right? So the companies make money as quickly as they can go and have a large patent life on their drugs. So whatever time you spend in development is lost time in terms of patent life. The clock starts kicking when you apply for the molecule early on. And again, the people in charge, they want to press you for that. So let's do that quickly, let's do that quickly. And that's really not conducive for you to try to learn and adapt and experiment. So people have to think more, as Chris was presenting, in the context of the program, not just in the context of the trial. So going quickly to failure not necessarily is a good way to do things efficiently. Anyway, until what time do I have? So that I can, okay, 15, okay, oh, that's okay. So at the end of the day, one approach that I have used and many of us in this room here have used is try to convince people by quantifying benefit and cost. Said, okay, we can talk about qualitative things like I'm doing to you right now, but at the end of the day, it's much better to present numbers and say that, okay, what is the value that running this trial with 6 doses instead of 2 and using an adaptive allocation will bring to the overall development program? And that's where the beauty and the benefit of simulations come in, is that you can look at different scenarios, you can look at different strategies, and you can, when you talk to those people, like I said, the decision makers, and give them numbers and you make a compelling case, you can do a lot of convincing. So it's not that frustrating in that regard. People listen, right? But if we just try to go, well, that's the best thing to do because it makes scientific sense, not necessarily they will fall for that. So quantification is key, and running simulations, looking at scenarios is one way that people have to do that. So what I'm going to talk about is an example of when that was done, and that's this adaptive dose ranging studies working group. So that's a group that was formed by PhRMA, this trade association of pharmaceutical companies in North America, mostly in the US, back in 2005, and it was part of a bigger program, which was a reaction of industry to the critical path document that FDA released in 2004. So the industry felt like, we should not be behind. Let us take the initiative and see how we can address our own problems. So that was one of the that came out of it. So there were several of us here, including Frank, Bjorn, Vlad came later, and Mike from the very beginning, that were involved in activities, and Beat also was involved in the traffic. So anyway, the purpose of this specific working group was to investigate methods to better learn about dose response. So from the early on, say, well, how can we do it better? So how can we try to quantify it, and how can we try to demonstrate its value for the broader community? So that was the intention. And how did it, so we are, for the most part, statisticians, with the exception of two of us, Mike and Rick, who are clinicians. So we had a strong bias toward quantification, looking at operating characteristics, running simulations, and that kind of comes through in the papers that we presented. But at the end of the day, we say, well, let's get a bunch of methods, right, that have been proposed, or that at least some of us here are able to code, and let's make a comparison. Let's write a protocol for the simulation piece so that we all do, in principle, the same. So we all have the same set of assumptions, and try to get the methods together and compare them also on a pre-specified set of operating characteristics. So that was the idea, right? Very simple. The focus of this working group has been not only on adaptive, even though the name is adaptive dose ranging, but that's a sign of times, right? Adaptive is one of those buzzwords that were very compelling at the time, even though the name, well, anyway, that's a digression. But we also looked at model-based methods. So some of the methods are not adaptive at all in the sense of changing allocation. Our group from the get-go has also been very much close to the adaptive designs working group, which is a different, that's the one chaired by Mike Rams back in the room, which was focusing more on higher level recommendations about the use of adaptive design. So our was very focused and very much limited to a certain class of methods. Now, Sujane, thank you very much, by the way, Sujane, for all the support that you have given over the years. Sujane has been a discussant of both the first round and the second round papers, and because of her support, I think this paper's got a lot of traction, and I really want, on behalf of the whole working group, I'd like to thank you for that. So there was a first round in which we looked at a set of methods, but there was a problem with it, that only two of those methods were indeed response adaptive in the context of changing allocation. But the key component of it was to look at operating characteristics for three different things that are typical in phase 2 trials. So the first one is just to detect a dose response signal. It's like a proof of concept component, right? Which is, it's essentially like a hypothesis test in that regard. So do I have something or not? Most trials would have to either address that in the trial itself or break that into a 2A component, and then in the 2B, you would look at the other two elements, which is selecting a dose for phase 3 and ultimately estimating the dose response. So these are an increasing level of difficulty when you look at them, right? And the key conclusions and recommendations were published in a white paper that got some nice traction. This is just like the first page of it. And two of the leading authors are in the audience. Where is Bjorn? Oh, in the back. Well, he was here. Oh, there he is. So that appeared back in 2007. So there are a lot of people, 5 of the people here in this author list are in this audience right now. But there was a second evaluation round. So that's what I want to focus a little bit more on with the 10 minutes that I have left. Because there were some issues when we presented and discussed this, especially at FDA. There was a very good meeting at FDA in which we showed the results and we say that, oh, this is what we think. These are some recommendations. What's the reaction? And they say, well, that's fine, but you guys should go back and look into other things that were not quite explored in the first round. The first one was obvious, is that, okay, we only had 2 adaptive methods in the first, so well, look at more and see if that kind of look at more treatments in a way. And we did. So Vlad actually chaired this particular work stream, and there were like 5 additional methods, adaptive, response adaptive, that were considered. And so Jane mentioned them by name and the people who were involved. There was another one that was, I think, interesting, led by Zora Antonijevic, that said, okay, what does it matter, the choice of those in phase 2? And that's a lot what Chris is exploring. So does it really make a big difference if I get it right or wrong? What is the impact of the dose selection, how much you invest in phase 2 on the phase 3 part? And the third one, which was quite interesting, led by Amit Roy, was to say that, well, can we do better by, instead of looking at the dose, looking at exposure, right? So maybe a lot of the problems that we face is because we're trying to put everybody on the same. So just looking at dose ignores a lot of the patient-to-patient variability. Look at exposure, maybe something better. So that was intended to evaluate, so if I were to use exposure response instead of dose response, how much value can that bring to me? So again, I have very summarized results, and I won't have time to go into details, but you can recognize in here the methods that Sujane was referring to. So this is adaptive MCP mod, Bjorn and Frank led that, Vlad and Krishna. But anyway, you got the idea. So there are like 5 methods, and there's a placebo or standard of care, which is a ANOVA, not placebo, that's that. But I mean, when you look at this, there are there's a lot of information, right? I don't want to go into that. So the bottom row is for 5 doses, and the top row for 9 doses. And the different columns refer to different models, the different scenarios that we consider. But perhaps the most striking feature in here is the lack of, right? So say, okay, well, I don't see anything. I don't see a color appearing throughout as the best. So by the way, what you look in here, some probability of getting the dose right. And again, I don't want to go into details what it means, but the higher the better. So you're talking about probability of making the right decision. Well, sometimes there's a method that comes on top, but maybe other times that method is not doing so well. So it's really dependent on what the model that you assume. Like in some cases, ANOVA doesn't do that bad. In others, it does a very lousy job. So it's really dependent on what you are considering. Something else to consider is that as I increase the number of adaptations, the number of interim analysis, there's kind of a leveling off. So most of the methods, there's a diminishing returns on investment in that regard. But there's not a method that comes up and say, aha, this is the way that we should be doing dose response, adaptive dose response. All right. This is a different component now. So I'm looking more at the selection of the dose. Now I'm considering the estimation of the dose response itself. So you're talking about prediction error in here. So the lower, the better in that regard. And then you start seeing something interesting going on, is that some of the methods, this one and this one, which happens to be the adaptive MCP mod and the t-test, they tend to get a bit worse as I increase the number of adaptations, which is, well, why is that going on? Well, there's an interesting aspect of it, is that both of those methods, the way that they're written, are focusing on the estimation of the right dose, not on the estimation of the dose response as a whole. So they try to zero in on the dose that's going to be best by detriment of not placing doses throughout the method to get a better estimate of the dose response. So that actually, the conflicting interests that you might have does explain partly the behavior that you see here. But again, you don't see a pattern coming up throughout. There are situations in which ANOVA actually performs okay, others that it's fairly lousy. So again, lack of percent. Anyway, I think I'll probably skip the exposure response, but this is, by the way, where my Greek letters appear. That's the only place. So I won't have a chance to talk about it. So I do want to talk about this because that was interesting when Zoran and others were working on this. And this is comparing now, evaluating the impact of what you do in phase 2 in the context of the phase 3. So for this particular exercise, we use the methods that were used in the first round because we already had a simulation. So we just want to see, well, if I were to get those results and apply and bring it to phase 3, what kind of power would I get? What kind of probability of success would I get in my phase 3? And in particular, what we were trying to single out was a recommendation that came out in the first round, which is that we should bring 2 doses forward into phase 3. So I recall that. I think that speak to FDA. The FDA, like people like Bob Temple, Sujay, and Bob O'Neill, have been talking about that for quite some time. So people in the industry were not too happy about it, but so we said, well, let's try to investigate that, right? So what we see here, again, is the average probability of phase 3 success when you bring 1 or 2 doses. Well, I guess it does make sense that if you bring 2 doses, there's a larger sample size. One would expect that to happen. But a more interesting question is this one, is that, all right, if I take into account that I'm spending more in my phase 2, that I may take more time to run that phase 2. So if I look at the bottom line of the company, which is the expected net present value. And for this one, Parvin from Wyeth at the time, Pfizer now, was crucial because she had some financial models that allow us to actually evaluate that in a sort of interesting and realistic way. This is for neuropathic pain as an indication. And in doing this, we still see, almost didn't screw up in the last one I did. We also see this very clear pattern of this red dot, which corresponds to 2 doses. Accelerated here means that we are investing more money to run the phase 3 trial with 2 doses at the same time that we would have with just 1 dose. We just pay more money, right? So that we don't want to waste patent life. But throughout, and that's kind of, even though the magnitude varies a little bit, but it's consistent that going with 2 doses does pay off for the assumptions that we have considered. And again, I like to highlight that if I change my assumptions, I can make it in such a way that probably 1 dose is going to be better than having 2. But under the assumptions that we chose, which were not intended to show this, they work quite well. So these were like interesting ideas. But at the end, in the second round, the group put together another white paper. And these were some of the key conclusions that had come out. First one is that, unfortunately, there is no single best way of doing things. And that's truth, I think, of life, unfortunately. So say that, well, that's it. So it would be nice to have this method here is going to give you the best dose-finding way of doing this, but there's not. There's not a design or a method that uniformly beats all the others. It depends on the assumptions, depends on the scenarios, depends on the conditions that you place. Another important thing that, of course, it's obvious, but we would like to just comment that, is that you're not talking about just adaptive designs. It's adaptive plus model-based or Bayesian or involving optimal designs, some combination thereof. So when looking about that, it may be interesting to try to evaluate that almost as a factorial experiment. So what is this impacting? What is that impacting? Where is the real value coming from? Learning priorities in the trial may lead to different bests. So if you are focusing primarily on getting the right dose, you may have to sacrifice some of your estimation of the dose response and vice versa. An idea that was not natural at all to me when I started this many years ago. So, well, if I do one, the other one is going to fall naturally. Not the case. So if you may want to allocate resources, like at the CRM in some sense, you may want to allocate resources just to the dose that matters to you, and you don't care that much about the whole region. Other times, you really want to learn about the dose response. It's like going D optimal, C optimal, I guess. That would be the... but there are better methods of doing than what we currently do, which is this pairwise comparison to placebo. So in all this, let's say, not so encouraging news, it is better to use model-based. It is better to have Bayesian methods. It is better to do adaptive, definitely so. But the magnitude of how better it is, it's going to vary. So 5 minutes? No, 2 minutes? Or yeah. Okay, just kind of, okay, almost done. So in terms of recommendation, I think that's consistent with that the working group put together is that, okay, instead of trying to come up with the method that should be used is this one, we say that, well, what we should be considering is this toolbox approach, right? So what we as quantitative scientists, like modelers, statisticians, and others in this field, should not just be thinking about one thing that is going to try to make this better, but rather to consider response adaptive, model-based, optimal designs. And in many ways, this conference talks about, I think, all of those in different chapters. And it explores the response as well. Revisiting the resource allocation between phase 2 and phase 3 is an important, it should be on the table, right? So if anything, what I see in my practice is that phase 2, people try to get it less and less important and faster with fewer and fewer doses. And I, even myself, have seen, like in my career, many examples in which companies pay for that dearly at the end when they go to get approval, FDA or EMA is going to come back, well, very nice, but go back and redo your phase 2, and then you lose another 2-3 years of patent life because of that, and that's very expensive. Now, software, many of us have talked about that, is absolutely critical for that. So we can talk all we want about methodology, interesting things. If that's not implemented in reliable software, it's going to be a nice publication, but it's not going to make it in the real world. So having something that actually implements that is absolutely fundamental, and we put that forward as a recommendation that open source would be the way to go, recognizing that that may not be possible in many cases. But there are some encouraging examples, not open source, but at least free software that's coming out of high quality. I think I'm just, just to mention here very briefly that the white papers from the second round have been published in Statistics in Biopharmaceutical Research in November of last year as a special issue. So just to leave with you with some further thoughts, and again, some of those are coming from the first round of evaluation. It's an obvious thing, but one that I think has led to a lot of problems in the industry. It's like the realization that detecting those responses is a lot easier than estimating it, right? Sounds obvious, right? So getting some signal is much, much easier than saying, what is that signal? But for the most part, what we do is that we power studies, we design studies based on the first one, assuming that the second one will come naturally. So I think for a clinician, oftentimes it's very hard to grasp the concept that if you have 95% power to show that you have a dose response, you may have a very lousy trial to getting you the right dose to go into phase 3. So the two are completely, I wouldn't say unrelated, of course they are not, but they are not equivalent by any stretch of the imagination. So the sample sizes that we use for phase 2 as a consequence of the first one typically are not sufficient, right? So you get not enough patients to really even try to, or enough doses for that matter, to try to explore what the dose response is. So those methods like adaptive, model-based should be considered routinely, but at the end of the day, if we don't change this mindset of phase 2 on the cheap, things are not going to happen. So we may be seeing, well, this method is better than that, or this approach is better than that, but both are lousy because my sample size is very small. So you may be optimizing between things, but the restriction that you face are such that it doesn't matter what the optimal is. The optimal is not nearly enough to be good for the ultimate goal of you being successful in getting the drug to the market, right? So this should be something that industry, regulators, academics should be doing together. So methodological development needs to be there. Industry has to embrace it. Regulators have to make incentives. So this is how the economy of drug development has to work, and this has to change. So sorry about overrun.
Thank you very much.
Thank you.
So, like I said, we will have plenty of time for discussion, but if you have some questions for clarification, we can take one or two simple questions. Everybody is hungry. Thank you.